Welcome everybody, and thank you for attending today's webinar. During the webinar, please send your questions by private chat to me, John McKinley, the host. They will be answered at the end of the webinar. Cartera is helping many of our customers screen libraries of antibodies in the race to find cocktails of therapeutic antibodies to combat COVID-19. We also have a new nickel biosensor chip for the LSA here at Cartera. And today, we're fortunate to have Tom Yuan from Twist Bioscience present on the success of their antibody engineering platform and workflow in their fight against COVID-19. Tom received his PhD from the University of California, Irvine, under the mentorship of Professor Gregory Weiss. Tom was an early scientist at Surrozen, where he developed bispecific antibodies as Wnt agonists in the regenerative medicine space. Tom then joined Twist Bioscience in 2018 as part of the biopharma vertical to drive antibody discovery and optimization by leveraging Twist's unique DNA synthesis platform. Tom, I'll hand it over to you now.
Thank you, John. I hope everyone can hear me nice and clear. I'd like to thank Carterra for inviting me to this webinar, and of course, everyone for spending the time today to view this webinar. Today, I'm just going to be going over some of the preliminary data that we have in some of the antibody drug discovery efforts where we're looking at for fighting the COVID-19 pandemic. Obviously, this has had a huge effect on all of our lives, and it's something that we've really pivoted towards in the last couple of months. Admittedly, we've actually got quite a late start to this. I'll go over this in the webinar, but everything that you see here is actually compressed to about a 6-week time span from the creation of some new libraries specifically targeted towards anti-SARS-CoV-2S1 spike protein, all the way to reformatting to full IgG to some of the initial kinetic characterization that we have here. So Twist, if you're not familiar with Twist, Twist Bioscience is the parent company. Their main thing is DNA synthesis. So their core technology is basically the silicon chip platform where they're able to make up to 96,000 genes on a single chip. Typically, if you do oligonucleotide synthesis, it takes an entire 96 well plate to make one gene. At Twist, the scale is several orders of magnitude higher, which is why they're able to provide many of the oligonucleotide orders, clonal gene orders at a much cheaper price. The department that I'm in is called Twist Biopharma, and Twist Biopharma is essentially started under the mentorship of Aaron Sato, mainly to leverage this DNA synthesis platform for antibody discovery and antibody optimization. So the primary way that we use this is we will use the print the oligonucleotide pools to create antibody libraries. So for example, in a typical antibody, let's say in the heavy chain, you have the constant framework regions in green, and then the variable CDR regions in these other colors. Typically, the way we design these libraries is that we specify the exact CDR sequences that we want to insert into each domain. And then these get mixed combinatorially so that we have, we normally try to target within a variable heavy or variable light, about 10 to the 10 in terms of the theoretical diversity. This contrasts to a more traditional mutagenesis approach where I'm using NNK or TRIM to assemble your antibody libraries, where you can't precisely control the exact sequences that are going into your library. When we design our libraries that are either fully synthetic or semi-synthetic, you print the exact sequence that you want for CDR1, and then you can designate exactly how many of those CDR1s you want. They can also vary in length. And you know exactly what's going into the library, and we also NGS sequence our libraries to make sure that the output of the library, once we've constructed it, is exactly matches the initial design that goes into it. So you can rationally sample your desired sequence space. Obviously, when you're printing the exact sequence, you can remove any amino acid liabilities such as isomerization sites, cleavage sites, deamidation sites, glycosylation sites. So the initial library that you're putting into does not have these to begin with. You can also do things like include specific motifs that you want or exclude specific motifs that you don't want in your antibody library. So what does Twist offer with this silicon-based DNA synthesis? Like I mentioned before, you have exact, you can avoid specific restriction sites. You can vary the length of your CDRs. You can create libraries that specifically ratio control the amino acid distribution at each site. Alternatively, you can literally select the exact sequences that you want to put in as well. And you can also include multiple germline scaffolds. And like I mentioned, we verify our libraries by NGS. So the way that we've been tackling this, both this project and also other projects, is that we don't use a single library. We use multiple libraries to target any either soluble antigen or cell surface antigen. We have specific libraries that we've developed tailored around targeting GPCRs, targeting ion channels. We also have another separate platform to optimize and humanize that we call TAO, the Twist Antibody Optimization Platform. And we've actually, as we've developed the lab, we've also generated, our protein sciences team has done a great job and generating a new workflow for high throughput antibody production. And what this allows us to do is, once we have the initial leads and our phage display libraries, we can convert or reformat to IgG hundreds of antibodies at a time. And we typically do this at a 1.2 mil scale. And that actually gives us enough protein to do the initial kinetic characterization, some epitope binning, and even some competition data just from that 1.2 mil transient transfection. And this actually plays well with Cartera as well, because the Cartera is a great system for assessing hundreds of antibodies at a time. But the way it prints these antibodies is great because you can have a very low concentration, say 5 or 10 micrograms per mil, and you actually recover most of your antibody when you print them onto the chip, which is actually great because once we print them on the chip, we can use the chip over and over again to assess kinetics. We can use it to assess epitope binning, but then still retain that IgG and do some other downstream assays. So this is just a slide kind of overviewing some of the libraries that we have on hand right now. One of our big focuses is with actually single domain VHH libraries. We have several different forms of this. We have libraries that target against ion channels, carbohydrates, GPCRs, and also libraries derived from what we call hyperimmune libraries, where the CDRH3 will have actually millions of different sequences. So what's a little bit different about Twist is that we can actually generate these libraries and make these fit for purpose libraries within an extremely short amount of time. And also the discovery cycle is fairly short as well. So our typical workflow is around 8 weeks. After we design the actual the antibody libraries, we are primarily putting them into a phage display vector. So in this case, we're displaying small fragments of the antibody on bacteriophage, and the bacteriophage gives us that the bacteriophage is what we're using to actually pan against or look for binders for a soluble protein or cell-based targets. This typically takes about 2 weeks. And once the panning and screening is done, we sequence all the positive hits that we have, either by NGS or Sanger, depending on whether it's a soluble protein or if it's a cell-based target. It then takes us roughly about 3 weeks to reformat to an IgG and DNA scale-up. So this is a little different because we do some initial hit picking in the phage display format, but all of our screens are primarily after reformatting into the full IgG, because obviously we want to test the final product. We're not testing antibody fragments. We're testing the final product after it's been reformatted to say IgG1 or IgG2 or a VHH-FC. This then goes through high throughput IgG purification. It takes roughly a week to do this. And then from this, we use this plate to conduct the binding and functional assays. Typically for our high throughput IgG for 1.2 mil scale, we get roughly 100 to 200 micrograms of usable protein or usable antibody, which is enough to conduct the initial kinetic characterization on the Cartera LSA. So this slide is going over some of what we call our high throughput antibody protein production. We're actually planning on introducing this as a new alpha product where you're able to actually submit your antibody sequences that you want to express and purify, and we will go ahead and express these for you at either 1 mil scale or 8 mil scale, purify them by protein A, and deliver that purified antibody in either a glycine buffer or an amine-free sodium acetate buffer. So we can do this with IgGs, single domain antibodies. Obviously, Twist already delivers purified DNA, but we're able to supplement this with the purified antibody. And like I mentioned, this is a Thermo XP293 transient system. Some of the metrics we can apply, introduce as well, or provide as well as obviously is digital SDS, percent purity, yield, and some additional optional tests as well, including affinity assessment by Cartera. So looking at the COVID-19 pandemic, one of the recent publications, I'm sure if you've been following this, you'll see that ACE2 is, or human angiotensin converting enzyme 2, is one of the primary cell surface receptors by which SARS-CoV-2 mediates cell entry and infection. TMPRSS2 is also required. This is a serine protease that cleaves, I believe, the spike protein, which then mediates the infection by the ACE2 surface receptor. But this paper actually looks at how SARS-CoV-2 is phylogenetically related with some of the other known coronaviruses. So if you look at by phylogenetic tree, you see that the SARS-CoV-2 clusters with the S proteins of known bat coronaviruses. And then if you look at the actual sequence of SARS-CoV-2, the spike protein S1 here is denoted here. RBD is the receptor binding domain. RBM is the receptor binding motif. And they're able to see just by a sequence alignment that ACE2, that it's highly similar to the bat coronavirus S1 proteins that are able to bind ACE2, and there's low similarity with the bat coronaviruses that are not able to bind ACE2. So this is sort of the first indication of what the actual cell surface receptor was that mediated viral entry. This is another paper published in Cell that actually looks to see if the introduction of soluble ACE2 actually reduces the amount of internalization that you see from the SARS-CoV-2 virus, and they're able to show that when you introduce soluble human recombinant ACE2 in solution, that you're actually able to lower the SARS-CoV-2 internalization, which suggests that human ACE2 is the primary receptor by which the coronavirus mediates cell entry. So we're able to use this and try to tackle and reduce SARS-CoV-2 infection by two different mechanisms. One is by targeting antibodies against the S1 protein itself on SARS-CoV-2. The other is actually looking to see if we can block viral entry by blocking the ACE2 receptor on cells, and by blocking the ACE2 receptor, it should be able to block the binding of SARS-CoV-2 to ACE2, and then thus decreasing the amount of virus and viral internalization. So what we've done is, so actually before then, this is just a cryo-EM structure looking at the pre-fusion complex of the spike protein. And if we look at exactly where the spike protein binds to ACE2, we actually see that the RBD domain actually binds away from the catalytic site of ACE2, which is important because we want to make sure that so we want to make sure that we're not affecting the catalytic site, or possibly not affecting the catalytic site if we design antibodies that block the S1 RBD binding interaction with human ACE2. So we've also announced a collaboration with Vanderbilt University, where we've partnered with them to design a synthetic library based on sequences from a convalescent COVID-19 survivor. The idea behind this is that if you have a COVID-19 survivor already generating antibodies, we're able to take some of those sequences and then design a new semi-synthetic library based off those sequences to potentially target and more effectively target the SARS-CoV-2 S1 spike protein. Some of the libraries that I'll be showing in this webinar actually have sequences derived from this library. So this slide is going over the general overview of the targets that we in terms of the entire workflow. So the two targets that we're looking at in terms of finding antibodies to bind to are the SARS-CoV-2 S1 RBD domain, and then also the ACE2 ECD. We're using multiple different libraries of both scFv format, VHH format, and each of these libraries are over 10 to the 10 diversity each. In terms of a phage display panning, we typically pan about 3 to 4 rounds. What this means is we're taking these antibody libraries, exposing them to either SARS-CoV-2 S1 or ACE2, allowing them to bind, and then washing away the non-binders, taking that pool and enriching that pool, and then repeating that 3 or 4 times over. And the idea is each time you repeat the rounds of panning, that you'll go from a large billion member antibody library, and by round 3 or 4, you're narrowing that antibody library to thousands of clones, but you're enriching the amount of binders that actually bind to your antigen of interest. Typically, we screen for about 1,200 separate colonies for each panning round to make sure we capture as much diversity as possible, and then we go into IgG cloning expression, and like I mentioned, we use the high throughput IgG product offering, the alpha offering, actually internally for ourselves as well, to produce enough antibody for kinetic characterization. And for testing, we look at affinity, we look at the receptor ligand competition, and also we look at developability to make sure that these antibodies are not, for example, thermostable. They're not sticky. They're not, they don't bind to everything. We see specific binding to either ACE2 or the SARS-CoV-2 S1. And like I mentioned, we've actually compressed this time scale where we're going from the library design to actually having some initial hits in hand in about a 6-week time frame. Obviously, if we also threw in some existing other antibody libraries, not just the COVID-2 phage display library, but also our hyperimmune phage library, and also our single domain phage libraries to see what we can extract out of these. So this slide is showing the phage selection rounds and titers. Like I mentioned before, when you're doing a phage display campaign, the goal is to enrich for antibodies that bind to your antigen of interest. So in the first initial rounds, you want to be gentle with your stringency so that you don't wash away potential binders that have, say, very low copy number. in the first round. As you go through the rounds, you want to increase the amount of washes, so you're increasing the amount of stringency, and you're lowering the amount of antigen that's present, so that you're further enriching for productive binders. The numbers here in bold for the different project codes indicate the phage titers. So what this is showing is that as you're going through each round, you want to see this phage titer slowly increase. Basically, it gives you a first initial peek as to whether your phage selection is actually working or not. If you're not seeing them increase, typically what you're doing is you're enriching for non-functional binders, or you're enriching for antibody clones that happen to express faster than other clones. But if you see this nice increase, say for example, from 2 times 10 to the 6th to 1.4 times 10 to the 8, then this is a strong indication that you're getting productive binders to the antigens that you're of interest. So this is some of the early, the Phage ELISA data, just demonstrating that with these antibodies expressed on phage, that they actually bind to either SARS-CoV-2 spike S1 protein or the ACE2 protein. And you can tell that from, just from the ELISA data, that we're getting hundreds of potential hits in terms of looking at the OD of fold over background. So what we did was we took all these ELISA positive hits, sent them in for sequencing, and once they're sequenced, then we reformatted them into a full either IgG1 for the S1 anti-S1 antibodies, or an IgG2 for the anti-ACE2 antibodies. This is looking at just a phylogenetic tree, looking at both the ELISA data, and we also did a crude binding inhibition study, looking at these anti-SARS-CoV-2 S1 binders and whether they're able to inhibit binding to ACE2. This is like a rather crude assay, but it gives us some initial hint before we spend the 3 weeks converting these antibodies to see if they have any potential to block the S1 ACE2 interaction. So what you'll see here is that bars that are deep blue show that they have potential high binding inhibition with ACE2. If we reverse this assay for the anti-ACE2 binders, we see that some of these clones as well have high inhibition binding, or in addition of the phage, gives you high inhibition of that ACE2 S1 interaction as well. And by looking at the phylogenetic tree, you see that this binding inhibition does not cluster around a highly similar family of antibodies. There we actually see a nice distribution in terms of these potentially antibodies that potentially inhibit the S1 ACE2 interaction. So we also did, before we converted these to the full IgG, we also did a specificity study looking at where exactly on S1 they did bind. We see that we do have a nice distribution of some antibodies that bind to S1, some bind to S1, specifically the RBD domain, and actually in our case, almost none of these bound to the RBM domain itself. So for where the Cartera LSA really comes in is because we already have such a high throughput workflow where we're not picking, say, the top 10 or top 20 leads, we're taking 100, 200, 300, 400 antibodies at a time and converting all of those. The LSA itself actually is really useful for us because it allows us to immobilize and print up to 384 antibodies onto a single chip at a time. So on the chip itself, the Cartera LSA has 2 main print heads. There's a 96 channel mode and a single channel mode. So like I mentioned, with the 96 channel mode, this is how you're actually, in our case, we're printing the antibodies on the chip in the 96 well mode and printing them in a grid of 4 so that you get a total of 384. Once the antibodies are mobilized on the chip, then we switch into the single channel mode and then flow over, say, the S1 protein or the ACE2 protein, usually with increasing concentrations to get the actual kinetics, the receptor binding kinetics. In terms of the LSA platform's core applications, we're mainly using the kinetics and affinity. I don't have the data yet in this specific webinar, but we are planning on running a full epitope bin on all the anti-S1 and anti-ACE2 proteins. And the LSA can also be used for epitope mapping and the quantitation as well. In terms of the 96-well printhead, typically what it will do is you have a plate. The 96-well printhead will take up your antibody and then flow into the chip back and forth. So what's nice about this is that, say you have a low concentration of antibody, say 5 micrograms per mil or 10 micrograms per mil, because it's flowing the reagent over back and forth, left and right, you can increase the amount of time in which you're allowing the antibody to immobilize without increasing the volume that you need. So typically for this, you need about 200 microliters to do the print, but your concentration can be fairly low, and you can just increase the amount of time to immobilize to increase the total amount of antibody that's bound to the chip. In terms of the single channel flow side, so once the antibodies are actually coupled to the chip, the single channel flow mode is used for flowing over the same antigen over the entire chip at once. So if you have, say, your spike protein, you're looking at real-time binding for every single spot, all 384 spots on this chip at one time. So getting into some of the data, it looks like we have actually a good number of picomolar level and low nanomolar affinity binders for both ACE2 and also S1. So for ACE2, many of these are 0.2, 0.3, 0.7 nanomolar. For S1, there's a few that are picomolar level, but there are many that are low nanomolar. And if you look at the binding kinetics, this is also very apparent, where you see a nice K on, and then you see a very, very slow K off for many of these clones. We also tested this against S1. We also tested against a full-length S trimer protein that Acro offers as well, to make sure that it actually binds to the trimeric form of the full S protein. And we see that in many cases that the antibodies that are able to bind the S1 monomer are also able to bind the full-length S trimer protein. Obviously, with these, you see that the K-offs are even slower, but this is benefits from avidity because it is a trimeric complex. So conversely, we also see that for the ACE2 binders, we have many picomolar level binders as well. For example, TV183-3, we see a KD of around 326 picomolar. Some of these, we also have a nice distribution as well. So they're not all picomolar level binders. There are ones that are a little bit weaker, single-digit nanomolar, double-digit nanomolar. But this gives us a nice kinetic diversity in terms of trying to develop these antibodies. One of the things we're actually also worried about is that ACE2 can be a rather sticky protein to assess. So we want to make sure that in our assay that it doesn't bind to, say, a non-binding antibody. So we put in trastuzumab just to confirm that the ACE2 binding that we're seeing here is not from some stickiness where the ACE2 is just binding to the chip overall. And this is also obviously apparent because it's not the binding kinetics are not exactly the same for every single antibody. This is some additional data looking at just some more clones that have very low picomolar or low single-digit nanomolar binding to either S1 or the S trimer. And one more thing we actually also tried is actually trying to see if we can use the Cartera LSA for an inhibition screen. So typically, in this format, you can immobilize the S1 protein onto the chip, and then if you have just the ACE2 protein in solution flowing over the chip, you'll see that it should be able to bind S1, and you see that with these curves here. So these curves here, this thick band here is the ACE2 binding to immobilized S1. Many of these are antibody plus ACE2 premixes showing no effect on the ACE2 S1 interaction. And then there are a couple clones here that show very clear inhibition where when we mix in molar excess the antibody with ACE2, that it completely blocks binding to the S1 protein that's immobilized on the chip surface. We're not using this as a primary screen, but it's because we already have the chip, we already have the antibodies coupled with the chip and use that chip to assess kinetics. It's pretty trivial to also couple S1 to the same Cartera LSA chip and then flow over ACE2 as well. So we're able to do the kinetics and also the initial inhibition screening in one go. So this is actually a kind of a neat sort of new use case that we have for the Cartera LSA. We actually went back and redid this inhibition assay in ELISA format as well, just to see if there's a real, if our antibodies are truly neutralizing. So for the S1 RBD H2 inhibition study, we used an existing kit from Acro in a competition ELISA format. What this is showing on the left is essentially binding of a biotinylated human ACE2 to immobilize SARS-CoV-2 S protein on a maxi sort plate. So essentially, you're immobilizing your S protein on the plate, and then you're introducing human ACE2 that's biotinylated, and then you detect that binding via streptavidin HRP. So you're detecting specifically the biotinylated ACE2 binding to SARS-CoV-2 S protein on the plate. When you introduce an antibody in a dose-dependent manner, as you increase the amount of antibody, you should be able to see a decrease in the amount of biotinylated ACE2 that you see bound to the ELISA plate. So at low concentration, for example, there's not enough antibody to block that interaction, so you see almost all the biotinylated human ACE2 binding to SARS-CoV-2 S protein on the plate. At high concentration of the antibody, you see this decrease, and with the proper dose response, you see a nice S curve, and you're able to extract an IC50 out of this. So we did an initial ELISA competition experiment looking at some of our proteins here. It's a little messy because we're showing quite a lot of these antibodies, but what we're looking at here is essentially on the left hand of the X scale is low concentration of antibody, almost a 0.1 nanomolar. And then at the right scale of the X-axis is high concentration of antibody, over 100 nanomolar. And what you see for some of these is that you have actually a very good dose-dependent manner in terms of which you're inhibiting that interaction of the biotinylated ACE2 in solution binding to the immobilized S protein on these ELISA plates. So, for example, Acro also provides a neutralizing antibody that's shown here in these circles here, and you see that some of these anti-ACE2 antibodies are able to actually improve upon that and will give a lower IC50 compared to the Acro antibody. This is also another set here where it's showing some of these anti-ACE2 antibodies have, they drastically inhibit that ACE2 S1 interaction here with very low percent bound, almost to zero, and of extremely low IC50 for many of these anti-ACE2 antibodies. We're also planning on testing this with the anti-S1 antibodies as well. I'm probably mainly showing the anti-ACE2 antibodies here. one thing to note is that in the Cartera experiment that I showed with the competition experiment, that was some of the main ones that were inhibiting were 182-7 and 184-7 and 184-57, which are also shown to be highly inhibitory in the ELISA format as well. So that it's nice to see those two data correlate. So in terms of our lead summary, we have, within 6 weeks, we have totally reformatted some of these IgG leads using our high throughput IgG product offering. For anti-SARS-CoV-2 S1 protein, we have 59 leads. Actually, for the first set, we potentially might have more as well. For ACE2, currently we have 184 leads, both in IgG2 format and VHH-Fc formats. And we have some high affinity picomolar level binders, and inhibitors, but we also have a nice spread as well, so they're not all picomolar level. In terms of future activities, we want to test all these antibodies, fully test to make sure that they block that receptor ligand interaction. And we also want to conduct a full epitope binning using the Cartera LSA platform. We want to make sure we test some of these as tetravalent and monospecific and bispecific constructs, and also obviously do a full developability assessment. We want to be able to partner up with CROs to do in vitro functional testing, so the pseudovirus reporter assay and also viral neutralization assays in lung epithelial organoids. And like I mentioned, we do have additional VHH screens against SARS-CoV-2 S1 protein. So we're still continuing in terms of screening for these antigen leads. So in terms of ways to work with Twist Biopharma, we're not a drug development company, but we do work with many collaborators to either discover or optimize antibodies. So this slide shows you many different ways you can work with Twist, either with licensing libraries, or partnering with us to generate new leads, or partnering with us to optimize existing leads. And like I mentioned, the high throughput IgG production is a new alpha product that we're starting as well, and we use this product in-house as well to be able to produce all the antibody protein that we need. And with that, I'd just like to thank everyone on the team. This has been a huge effort, a really fast pivot and a huge effort, both within the Twist Biopharma department, but also with the libraries team and protein sciences team and all, many of the different functions at Twist that without them we wouldn't be able to either design and synthesize these libraries or test these at the throughput that we're able to do. And with that, I will take any questions you might have for this webinar. As well, Noah is able to answer any technical questions you may have about the Cartera. And thank you for your time.
Thank you, Tom. That was an excellent presentation, very informative. We will now open up a panel discussion with Tom and Noah Ditto, Cartera's technical product manager. Noah supported drug discovery and early clinical development for nearly a decade at Bristol Myers Squibb. He's an expert in label-free biosensor screening technology and is one of Cartera's longest-term employees. If you have any questions, please send them by private chat to me, John McKinley, the host, and we will answer them. Okay, I see some questions coming in. One of them is regarding the data, how you use the data from convalescent patients to create libraries, and how many of your binders came from those libraries compared to naive libraries.
So I can't get into specific details, but the essential idea is we take the antibody sequences from someone who's recovered from COVID-19, and we can take some of the CDRs and incorporate those CDRs into our synthetic libraries. in a combinatorial fashion. In terms of the percentage, actually most of our anti-S1 hits came from that library. So I would actually say the majority would came from that library.
Another question, Tom. As a user of other kinetic platforms, how is the Cartera LSA differentiating for you at Twist?
So the LSA has actually been pretty instrumental in terms of our workflow. Typically with some of the other, label-free SPR methods, we actually do have, another smaller SPR that, can only run two channels at a time, but we only use that for, just verifying binding for, something we already have. The LSA has actually allowed us to screen kinetics for our IgGs and get real-time kinetics as a screening tool, not just a tool to confirm binding, say after we've done an ELISA screen or after we've done a Phage screen. So it's completely, revamped our workflow.
Tom, what regeneration condition was used for the SARS-CoV-2 kinetics?
So the regeneration, in this case, actually, we've been using the thermal IgG elution buffer, which is commercially available. So you can use that. We've also used glycine in the past as well, glycine pH2. It seems to work well, and it doesn't, at least in our case, it doesn't degrade binding of the antibody to ACE2 or antibody S1 interaction.
Thank you, Tom. We have a question for Noah. What are some of the considerations when epitope binding antibodies against the spike protein?
Yeah, that's a great question. When you think about the spike proteins and kind of where we are with COVID, it's been a mad dash for everybody to generate the proteins and get them out commercially available. What we've seen, and this really holds true for any binding assay, but I guess it gets highlighted in the COVID scenario, is that the protein quality can vary highly between these manufacturers. Tom, in his examples, is using the Acro Biosystems spike protein, for example. I know Acro Biosystems, they do SEC mols, for example, they check purity, and that's critical for these binding assays to make sure that you have a really, really good quality protein to start with, because otherwise your binding data can be highly variable if you have heterogeneous kind of mixtures of the antigen species you're binding against. That's usually the biggest impediment to really making clearly discernible data sets. Somewhat secondary to that, obviously, is the valency consideration. The spike protein is a trimer. You could also use a monomer form as well, depending on what your aims were. The trimer form would be best suited for a premix type assay, and then the monomeric form or monovalent form would be best suited for a classical type assay, sandwich style assay. And probably the last thing just to note with these spike protein assays, and Tom was sort of hinting at that with some of the inhibition data they had. You can take that surface with antibodies arrayed and simply assess whether or not S1, for example, and ACE2 can interact in the presence of being bound to these antibodies. So within the binding exercises, it's a great way to demonstrate blocking profiles in addition to clustering the antibodies based on epitope.
Thanks, Noah. There's another question for you. What is the minimal concentration of antibody needed for kinetic analysis on the LSA?
I'd say it's kind of variable. Depends on kind of the antigen you're using and the surface, the antibodies. So we don't want to really say there's a hard and fast rule. But we've seen successful enrichment of concentrations down. So the actual working assay concentration down into the mid nanogram per mil range. So you can go quite low. Either one, you have very low expression, or two, you may just have not much physical material and need to dilute it to a sufficient volume for the assay itself. So you can be in the nanogram per mil range and run kinetics as just sort of a lower limit of the assay.
Thank you, Noah. Tom, here's a question for you. Is Twist looking to develop these leads on your own, or are you looking to partner them?
So like I mentioned before, we're not, a fully fledged drug development company. We are actually looking to partner with other CROs and other companies and organizations to both assess the therapeutic value of these antibodies. So yes, we're definitely looking for partnerships.
Thanks, Tom. Another question for you. How do you use next generation sequencing in your screening process if you screen 3 by 384 and then transfer the hits to IgG?
So but we use NGS for two functions. One is to, once the library is actually produced and put into the phage display vector, we use NGS to verify that the output of all the sequences that we designed are actually in the final library and there aren't any frame shifts or, aberrant mutations. So it's a QC step. The other way we use NGS is to track the enrichment of the antibodies as you go through the panning rounds. So as you go through rounds 1, 2, 3, 4 of your phage panning, the idea is you're enriching for binders. And what NGS allows us to do is actually quantitate which antibodies are actually being fully enriched over others. So that way, say if you have 300, 400, 500, 600 ELISA positive hits, it allows you to sort of prioritize which antibodies seem to be enriching in the panning itself by NGS. So it gives us some more data and it gives us a way to triage if we need to triage.
Here's another question. It looks like you reformat your hits to IgG and then do kinetics. Have you tried to include a tag in your library, for example, a V5 tag? And then you can use the LSA to directly screen your phage hits?
So we have not. Our typical, one of the reasons why we prefer reformatting the IgG is it just gives us a better, well, first of all, once you reformat the IgG, typically we want to develop these as full IgG therapeutics. So we're testing the final product or something close to the final product. And when you reformat a lot of these, you'll see some drop out where they don't retain binding once they be reformatted from the SCD to an IgG. That's an interesting concept though. It's something that we might look into depending, in terms of integrating some sort of tag. Our phage libraries do have his tags, but we can also use other tags to couple, for the antibody fragments rather than reformatting into the IgG.
Tom, have you tested these antibodies for binding to S proteins from related viruses?
Not yet. We're actually, that's one of the things we're going to be doing next. So we're going to be looking to see, look at the cross-reactivity between a binding to, SARS-CoV-2 S1, also the original SARS spike protein, and also MERS. We're also looking to, see if binding is retained for some of the SARS-CoV-2 S1 mutations that appear to be popping up now. So specificity testing is definitely next in the dock for us.
Is there an indication for functional superiority for antibodies that bind to monomeric RBD versus trimeric RBD?
We don't have that data yet. It's something we definitely want to see if there's a difference. In our case, it mostly seems like everything that binds to the S1 RBD also, everything that seems to bind to the full S protein trimer also seems to bind to the S1 monomers as well.
Was there an assay development done to determine which conditions work best?
If you're talking about the LSA itself in terms of, say, regeneration conditions, yes, we did do some regen scouting. Obviously when you do so for example, the competition experiment, you want to make sure that you have sufficient S1 immobilized to the chip to be able to see ACE2 binding. and also be able to premix enough antibodies such that you're well in lower access to fully block that interaction. So a lot of the scouting was just scouting different concentrations, making sure we had enough material to get a proper RU shift, but not so much that you're not able to block the interaction with the competing antibody.
How do you design your library based on the antibody sequences recovered from patients?
So I think this is similar to another question we had previously. I can't get into specific details, but the core idea is that you're taking some of the CDR sequences that in this library and then shuffling or mixing them with some of the sequences that we have that we know are coming from other human antibodies, so that we're creating a semi-synthetic library. So the CDR sequences are lifted from these convalescent donors.
In general, what's the normal signal over background ratio in your final round of phage library panning? Round 4? Is this target dependent?
So the signal to background ratio in the ELISA is basically looking at binding of the phage with the antibody displayed binding to either S1 or ACE2, and then looking at whether that same phage displayed antibody binds to a background protein such as say BSA or any other protein, and that gives us that's how we calculate our fold over background. Typically, our minimum for that is 3 times over background, but if you look at the data on the slides, there are many that are 5 times, 10 times, 20 times over background. So we knew that we had a lot of very high affinity binders just from the phage ELISAs. I mean, obviously, the phage ELISAs are pretty crude. They're more of a yes/no assay, but that level of fold over background did give us a hint that we had good picomolar, nanomolar level binders, which did play through once we converted them and tested them on the Cartera.
In your antibody selections, you have a low number of washes. Do these include an incubation step where the beads sit and buffer?
Yes, so typically we'll do this on the either by, so they're all soluble phase, all the antigens are biotinylated and coupled to strict cavity encoded beads. And we've played around with the wash conditions in terms of the number of washes. We've kind of discovered that if we run this on the Kingfisher automation, that it's, there's not a lot of carryover compared to when you're doing this by hand. So the number of washes looks relatively low, like 3 washes, 4 washes, 5 washes, but this is equivalent to doing it by hand, something like 3, 6, 8, 9 washes by hand. So it's more dependent on obviously the format in which you're doing the panning and also the exact mechanism by whether you're doing it by hand or Kingfisher. I mean, the primary way we track this is by phage titer. So if the phage titers are going up and they're not too high, then we know that we're on the right track.
Thanks, Tom. Antibodies against ACE2 sound less appealing than those against spike RBD. What are the proposed advantages?
Well, one potential advantage is that you have less risk of viral escape in terms of mutagenic escape. The virus is obviously mutating quite, the virus has the potential to mutate quite rapidly. And we have to be able to test and for binding. Obviously, for antibody therapeutics, one of the proposed methods is by assembling a cocktail of antibodies, so that you're binding to different epitopes on the S1 at the concurrently, so that if say one part of the S1 protein mutates, you don't lose that neutralization ability. If you have an anti-ACE2 binding antibody, I mean, obviously you have to test to make sure that it doesn't negatively affect, the actual catalytic function of ACE2, but you're less likely to have some sort of mutational event where you're no longer able to inhibit that interaction.
Thank you, Tom. We have time for one last question. How do you assess the risk of blocking ACE2?
How do we assess the risk of blocking ACE2? So we haven't looked at that yet. Obviously, we have to be able to develop or do additional assays to determine if our antibodies affect ACE2 function. I mean, it does look like just from the S1 RBD ACE2 interaction is that if we're just blocking that interaction is quite well away from the catalytic site. So, you know, there's a potential that we're not affecting function, but obviously we have to test for this. And, you know, we're looking for partners to be able to do this. We're not currently set up for something like this, this assay in our own lab yet.
Thank you so much, Tom. That was a great presentation. For those of you whose questions we were not able to answer, reach out to get those questions answered. And I want to thank you again, Tom and Noah, for your help on this webinar. And I hope you all have a great day.